Yeah, sounds good.
Okay. Okay. So, hi everyone. Welcome to Martlets Society and the Martlet Talk Season 3, Episode 1. And thanks TMS for giving me this opportunity to be today's moderator. And my name is Bingbing Ying, and currently I'm a postdoc at MIT. Today, we are so glad to have Professor Sarkar from MIT as our guest speaker and Dr. Fei, my colleague, as our co-host. So from this season, we changed our name from TMS Talk to Martlet Talk, and we hope you enjoy it. And we will have some new activities in this season, and we believe we can present you another exciting season, especially when you feel Zoom fatigue. And before today's talk, let's acknowledge today's the volunteer for today's events. Thank you for your contribution. And now let me introduce today's agenda. Includes 4 parts. So in the opening part, our present Dr. Zheng Ying, a postdoc at the Max Planck Institute, will have a brief introduction of our society. As we realize today, there are many new friends joining us today. And Zheng will also touch a little bit about new activities for this season. And then our co-host, Vivian, will introduce today's speaker, Professor Sarkar. And then Professor Sarkar will share with us the recent research progress from her lab at the MIT, and it will last about 40 minutes. After the talk, we will have a 30 minutes Q&A session, and Vivian will lead this session. Okay, now I will pass the floor to Zheng. Zheng?
Hi, everyone. So I'm Zheng, and in case you don't know well about our society, the Martlet Society is a nonprofit independent organization. So we aim to provide a platform for young scholars to build connections and to show their work. And currently we have several series of academic events. We have Martlets Talk for academic research and WDA Talk for to show you the world beyond academia, and TMS workshop for skill set build. And we also have special issue based on the request from our audience. And like previously, we invited the chief editor-in-chief of Matter, Professor Stephen Cranford, and also Professor Teng Li from University of Maryland. And starting from season 3, we also will introduce some new events, such as the research contest and some type of research contest, and you will have a chance to win some prize. And also we will introduce the workshop for faculty applications. And I know that many of you will be very interested. And if you are interested in our events, you can follow us on WeChat and on Twitter and LinkedIn, and also our website. And if you have any questions or you want to be a speaker or host, please don't hesitate contacting us through our email. And yeah, that's it. Yeah, thanks a lot, Bingbing.
Thanks, Zheng, for the nice introduction, and really looking forward to the following episodes. Okay, Vivian, now the floor is yours.
All right. So hi, everyone. My name is Vivian Feig. I'm a postdoc at MIT in the Brigham and Women's Hospital. And today it's my distinct honor to introduce our speaker for today's seminar, Professor Deblina Sarkar. So Professor Serkar is an assistant professor at MIT and the AT&T Career Development Chair Professor at MIT Media Lab. As head of the Nano-Cybernetic Biotech Research Group, her work is transdisciplinary and combines engineering, applied physics, and biology. Her research bridges the gap between nanotechnology and synthetic biology on the quest to develop disruptive technologies for nanoelectronic devices and to create new paradigms for life-machine symbiosis. So Professor Serkar has received numerous honors and recognitions so far in her career. To just list a select few examples, her PhD dissertation was awarded the Lancaster Award by UC Santa Barbara and was recognized by the Council of Graduate Schools as being one of the top 3 dissertations in math, science, and engineering throughout the USA and Canada. And in 2018, she was named as one of MIT Technology Review's top 10 innovators under 35 from India, and also received the NIH Pathway to Independence Award. So I'm extremely excited to see how Professor Serkar's already impressive career is going to continue to flourish in the coming years. And I'm really eager to hear her share more about her research accomplishments and visions with us today. So with that, I'll hand the stage over to Professor Serkar.
Okay.
Thanks a lot, Vivian, for the very nice introduction. It's a great pleasure to be here today with all of you and share my work. So I'll go ahead and share my screen. Can you guys see my screen?
Yeah.
Okay, sounds good. So yeah, as Vivian just stated, I actually started as an assistant professor at MIT about 1.5 years ago. So I started my career actually as a nanoelectronics researcher, and then I moved to neuroscience, did a very steep transition to study the brain. So what I will do today is basically tell you the story of my journey from nanoelectronics to neuroscience. And I will tell you about the amazing synergies that exist between these two fields. And I will give you one example of project from each of these fields. So let's get started. So all of you must have noticed how your laptop heats up after working for some time. The heat generated is so high that a while ago, I even cooked an egg on my laptop. So let us understand where this heat is coming from. Your computer has 1 billion switches in them, and in those switches, the electrons need to jump over an energy barrier to cause current flow. And when this barrier is high, very few electrons go from source to drain, and the switch or the transistor that you call it is off. And when you want to turn on the switch, we apply voltage to another terminal, which we call the gate. And that way we reduce the barrier height. Then more electrons can go from the source to the drain and the transistor turns on. So ideally, what we would want that this transition from off to on state is very steep. That means with application of very small voltage, we should be able to turn on the device. And since the power is the square of the voltage that we apply, so that can reduce the power. However, we do not live in an ideal world. So in an actual transistor, we need to apply a sufficient amount of voltage to turn our device on, because the current increases slowly as we apply the voltage. So this transition is not very steep. Now, a parameter that describes how steeply this current increases with voltage is called the subthreshold swing. And that can be easily defined as the voltage that is required to change the current by a decade or by tenfold. And the formula is very simple, is basically the derivative of the gate voltage that you apply with the log of the drain current. So as you can understand, smaller is the subthreshold swing, steeper will be the turn on characteristics, and that's what we want. So, but let's see what is the problem that happens when we want to make the transistor smaller and smaller. And you want to make the transistor smaller because you want to fit more and more of them in a given chip so that you can increase the functionality of the chip. But however, when you scale down the transistor size on the length dimension, what happens is that the electric field from the drain kind of encroaches into the channel and it reduces the barrier height on the source side. Now, in that case, the electrons from the source goes to the drain even when the transistor is supposed to be off. So that increases the off-state current, and that means it increases the off-state power. As you can see here, for a long channel transistor, this off to on transition, it's steeper, so that it has better subthreshold swing or lower subthreshold swing. But when you go to this short channel transistor, because the steepness of the current voltage curve degrades, this off-state current has increased as shown by this green curve here. Now in the world of electronics, you can think of rule of thumb is that thinner your channel is, smaller it can also be in the length dimension. And that is because it improves the electrostatics. So how thin can we make the channel? So ideally, actually, we can make them in the scale of atoms. The channels can be atomically thin. And those are the materials which we call 2D materials, because they're 2D because the third dimension, that is the dimension of thickness, is almost like negligible because they're atomically thin. So these 2D materials have several advantages compared to conventional 3D materials. So our technology right now is based on silicon. Silicon is a 3D material. So you can think of these 3D materials, they are more like potatoes. You know, if you want to make very thin slices of them, you have to cut them into thin slices, but they will not be super flat. They will have some roughness to it. And because of this surface roughness and quantum mechanical properties, that will also lead to variation in its performance. However, these two-dimensional materials, 2D materials, you can think of them like onions. And these layers, they basically can be peeled off just like layers of onions. And each of these layers can be atomically thin. For example, it can be 1 to few atoms in thickness, which is like few angstrom level we are talking about. And so their surfaces can be atomically flat. And because they come in layers and there is no chemical bonding between any two layers, as we peel them off, there are no dangling bonds at the interface. So that helps to improve the electrostatics a lot. Now, I believe most of you would have already heard about graphene. So that is the most widely known 2D material. However, the problem with graphene is that it does not come with something called a band gap. So band gap is basically the region in energy where the electrons cannot stay. So that's the forbidden energy gap. The electrons cannot stay there. But we actually manipulate this band gap in a transistor to have the barrier for the electrons to flow. And if we do not have this band gap, basically we cannot distinguish between the off and on state. So we actually need materials which are 2D materials, which are atomically thin, but also should have band gap. So we have to think about beyond graphene 2D materials, and these materials which have band gap are called semiconductors. And transition metal dichalcogenides are one such material. So they are also layered structures. And within each layer, transition metals and the chalcogenides are bonded together chemically. But between any two layers, there is actually no bonding. So they also come in layers, and each layer of this material can be atomically thin. For example, one transition metal dichalcogenide, one example is molybdenum disulfide. And a single layer of this material is only 0.65 nanometer in thickness. So this is about dimensional scalability. We can scale the length of the, of our transistors if we can make our channel thinner and thinner. However, that does not solve the whole power problem because we also have a fundamental limitations in power scalability. So suppose we have a certain barrier height H in energy for the transistor when it is in off state. And if we apply a gate voltage VGS to the transistor, we will reduce the barrier. The maximum we can reduce the barrier is by about the same, the VGS, if we think the unit is in electron volts. And since the electrons in the source, they are distributed according to the Fermi-Dirac distribution, which we can approximate with Boltzmann distribution for higher energy, the current will increase by exponential VGS by KT as you apply a gate voltage of VGS and reduce the barrier by VGS. This is how the current will increase. Here K is the Boltzmann constant and T is the temperature. And if you calculate from here, the subthreshold swing that you will get from this device at room temperature is 60 millivolt per decade. So that means a conventional field effect transistor, we cannot make the subthreshold swing lower than 60 millivolt per decade. And that's because of this fundamental thermal distribution of electrons that are present in the source. Now, what it means for a transistor design is that if you make a conventional transistor, in the very best case, its slope or subthreshold swing is basically 60 millivolt per decade. That means if you want a current on to off ratio of 10,000, that is 10 to the power 4, you need at least 4 multiplied by 60, 240 millivolts of voltage. Now, this is the very best case, but in a conventional design, the subthreshold swing will be even higher than 60. That means slope is even worse than that. So you would end up needing much more supply voltage. So if we really want to solve this problem, we have to have devices where the steepness is much lower than 60. And as I mentioned, that this number comes from the fact that these electrons are, distributed according to Boltzmann's distribution, which is very fundamental in nature because that's related to the thermal energy of the electrons. So if we really want to solve this problem, just using a novel material will not help. We'll have to think of some fundamentally new carrier transport mechanism. So what do we do? So here in this case, as I mentioned, the electrons jump over an energy barrier to cause current flow, and we are basically reducing the barrier height to make the electrons go from source to the drain when you want to turn on the device. So instead of making the electrons jump over the energy barrier, can we just make them sneak through the barrier like a ghost walking through the wall? Now you must be thinking, how is this possible? Because, if I'm just standing here, I cannot just go through a wall like that. So while classical physics says that it is not possible, quantum mechanics says that it is possible. And this is because the electrons have also wave nature, and those waves can penetrate through an energy barrier. So that is all good. Suppose here there is this person here who really likes low energy and thinks that, oh, I'll just tunnel through this, or I'll just go and penetrate through the energy barrier. The scientific term of this is quantum mechanical tunneling. So if he thinks that he would go to the other side of the barrier by tunneling, but there is a problem. So by the time he reaches the other side of the barrier, he would look something like this. And this is because the electron waves decay exponentially as it passes through the barrier. So that means that if we want to use this tunneling mechanism, the electron waves will decay exponentially. And so the current that we can get through these devices will be extremely low. And it would be even lower than the noise level of the transistor itself. So if we really want to develop efficient devices where we want to have effective tunneling effect, that requires very intricate design. So to have efficient tunneling and reduce this exponential decay of waves, we'll have to reduce the barrier height as well as the barrier width and have high electric field at the tunneling junction. And achieving all of this at the same time is challenging. And moreover, note that just having a steep subthreshold swing for a small region of current is not enough. We need to have a steep subthreshold swing or very steep slope for almost like 3 to 4 decades of current if we really want to reduce the supply voltage. And the International Technology Roadmap for Semiconductor, they set the requirement that we need to have this sub 60 millivolt per decade subthreshold swing for over 4 decades of current. So that is like about 4 into 10 to the power 4 on-off ratio of current. We should have this subthermal or sub 60 millivolt at room temperature subthreshold swing. So next I will discuss our device that we developed, which can actually overcome these challenges and achieve low power operation, and it overcomes this fundamental thermal limits in subthreshold swing with tunneling effect. And we call this transistor Atlas defect, which is a short form for atomically thin and layered semiconducting channel tunnel field effect transistor. So in the next few slides, I will try to explain how this device works. So for this transistor, we actually used the 3D material as the source. So 3D material is used as a source because here we can leverage the mature doping technology of 3D materials to create a highly doped source region. But the material that you used for the channel is still 2D in nature. And as I discussed, you need to make the channel thinner and thinner, and that helps us to achieve dimensional scalability. So that is why the channel material is 2D in nature. And moreover, this junction between 3D and 2D material is van der Waals in nature. That means there is no chemical bonding between the 3D source and the 2D channel. And that helps because whatever dopant atoms we used for the 3D source, that does not diffuse into the 2D material. So that helps us to create highly, very sharp tunneling junction and electric field profile. So the electric field at such tunneling junctions can be very high. And also, if we think what kind of 3D material and what kind of 2D material we should choose for making this kind of tunneling junction. So we chose germanium as the 3D material and molybdenum disulfide as a 2D material because among the commonly known 3D materials, germanium has a relatively low electron affinity and lower band gap, while molybdenum disulfide has higher electron affinity. So if we align those bands and look at the energy landscape of these two materials, when you put them side by side, you see that the height of the energy barrier for these two materials for the heterojunction that we create can be low. So these are the two materials that we use, but there have been a lot of more discoveries of novel 2D materials. So it is possible to use and basically mix and match between different kinds of materials to improve this tunneling height and reduce it even further. And so we have high electric field at the tunneling junction. We can reduce the barrier height, but how do you also reduce the barrier width? So to explain this, I have drawn here the band diagram. So for people who are not really conversant in this field, I would try to explain this very simply. So band diagram is basically the energy landscape of the materials. And here I'm basically drawing the band diagram going from the source, which is germanium, and going through the two layers of molybdenum disulfide, which is our 2D channel. And then we have the gate dielectric and the gate metal. So we are drawing the energy diagram along this z-axis here. And whatever region you are showing, we are seeing here in colors are the regions where electron can stay. And the white region here, what you are seeing is the forbidden energy gap. And so that means the electrons cannot stay in this region. So in the off state, what happens is that, so the green region here is the germanium, and this yellow region is the two layers of molybdenum disulfide that we used. And this blue region is the gate dielectric, and here is the gate metal. Again, you're going from germanium, two layers of molybdenum disulfide, gate dielectric, gate metal here. So in the off state, what happens is the filled region in germanium, that is the region in energy that is filled with electrons, they do not align with the empty region in molybdenum disulfide. So as you can see here with the black arrow, so there is no current flowing. And if you go higher in energy, we basically encounter the band gap of germanium. So again, not many electrons, no electrons are present here, so no current. If we go even higher in energy, we basically go into what we call the conduction band of germanium. But we have designed our devices such that what we call the Fermi level is in here. And since the number of electrons will reduce exponentially as we go higher in energy, the number of electrons present here is very low. So that means the current that we will get will be very low in this case. Now, when you want to turn on the transistor, what you would do is we apply voltage to this terminal called gate, and we lower this whole energy band diagram, we bring it down. And then we can align the field regions in the germanium source with the empty region in the molybdenum disulfide, as you can see here with the green arrow. So now the region filled with electrons are now aligned with the empty region in molybdenum disulfide. So then the current can flow and the device can turn on at this point. So as you can see here, the thickness through which these electrons have to tunnel is determined by these two layers of molybdenum disulfide. And each layer of molybdenum disulfide is around 0.65 nanometer thick. So it's only 3 atoms in thickness each layer. Since we use 2 layers, the tunneling barrier width that we get in this case is only 6 atoms thick, which is extremely thin. And so that helps us to achieve high tunneling current. Now, this is the TEM image of the germanium and molybdenum disulfide heterojunction, and germanium is shown here, and these are the 2 layers of molybdenum disulfide. And you see that apart from the germanium and the molybdenum disulfide, there is also a layer of germanium oxide. Now, this is a challenge because this germanium oxide will add extra barrier width to our structure. So this is a challenge, but this is also an opportunity for future, because in future we can actually passivate the germanium layer and get rid of the germanium oxide. So that can help to increase the tunneling current even further. And here we're showing the subthreshold swing characteristics of our device. And so the red line here shows the fundamental limit, that is 60 millivolt per decade at room temperature. And you're plotting the conventional field effect transistor characteristics, which are shown by these blue squares. And the characteristics of our tunnel field effect transistor, the Atlas effect, is shown by these green triangles. As you can see, for a conventional FET, for a conventional transistor, the subthreshold swing is always above this 60 millivolt per decade line. And as you can see, it increases, but it's always here at room temperature. However, we can see that Atlas effect actually beats this fundamental limit and leads to a minimum subthreshold swing of around 3.9 millivolt per decade. And if we average it over 4 decades of current, which is the ITRS requirement, we get an average subthreshold swing of 31.1 millivolt per decade, which is almost half of the fundamental limit. And apart from having this subthreshold swing that you overcome the fundamental limit, which allows us to lower the power, because lower subthreshold swing means that you can reduce the supply voltage, and power is basically square of the voltage. But apart from that, since this is also only 6 atoms thick, the channel, that also allows dimensional scalability. So 6 atoms is only around 1.3 nanometer in thickness. So the 6 atoms thick channel allows us dimensional scalability. So this device can achieve simultaneous power and dimensional scalability at the same time. And since you reduce the power, that's good for our environment. So that's why we call this like green electronics. So if you're wondering from the title of my talk, green electronics to gray matter, what it means. So low power electronics, good for environments. So that's why green. So while working on these low power electronic devices for electronic computations, I also got interested in other non-electronic computational systems, specifically the brain, because the brain probably can be thought of as the lowest power computer ever. So the brain, which is this amazingly complex organ and the boss of our body. And it weighs just 3 pounds, but it's really the brain that determines who we are and differentiates us from each other. And if we go inside the brain, you will see that it's formed of hundreds of billions of computational elements, which we call the neurons, which talk to each other using electrical and chemical signals. So the brain does all this computation, but actually consumes only about 20 watts of power, which is similar to a mere light bulb. So it's extremely energy efficient. However, at present, we know very less as to how the brain works. So why is it so difficult to understand the brain? So if we think of electronic circuits, we build them ourselves. So we know all the interconnections between all the components of our electronic chip. And not only we know the connections, but we also know each and every device, what kind of materials it is made of. We know their configuration, and we know the physics of how that works. So we can draw band diagrams for metals and conductors and insulators and explain the physics and their working principle. However, in the brain, Mother Nature has made it so complicated and so intricate. Not only we know how the individual components and computing elements are connected to each other in the large scale circuit, but even we don't know in a single neuron, for example, how the biomolecules are arranged and how computation happens. And if we look into a small region of the brain and zoom in, we'll see that it's formed of a dense jungle of biomolecules, and which would probably look very similar to a gum wall in Seattle. So if we really want to decipher this dense jungle of biomolecules, we need super resolution methodologies. However, the present super resolution technologies are either very complex or intricate and requires very expert handling and does not work in the scale of the whole brain. So in this respect, a technology called expansion microscopy is very interesting. So expansion microscopy is based on the principle that instead of using complicated hardware to see small features in the sample, can we expand the sample itself? So suppose there are 2 points which are very close together, and because of the diffraction limit of light, we will not be able to resolve them as 2 separate points. However, if we physically expand the samples and separate them from each other, we will be able to decipher them and resolve them. And so how do we actually expand a sample of biological tissue? So we can use in this respect some polymers which are called hydrogels, and sodium polyacrylate is one of them. So this hydrogel, when dry, looks like this, but when you add water to it, they will swell up and expand. So this is relatively simple in how we can achieve this. So suppose we take a piece of brain tissue and we embed them in this polyacrylate hydrogel, and then after some chemical processing that we need to do, we add water, and you see that the whole brain expands. And this is actually the same brain tissue that you see in the left. And on the left side is before expansion, and the right side is after we do about 4.5-fold expansion. Now, if we think of the diffraction limit of light is around 300 nanometer, after about 4.5-fold expansion, we'll get about 70 nanometer of resolution, still using the same diffraction limited microscope. So the hardware remains the same, but the resolution actually overcomes the diffraction limit here. So to tell you a little bit about how this process works, we start with a fixed specimen, and then we anchor the biomolecules of interest. So these anchors basically are used so that any biomolecules of interest, they can be connected to the polymer gel that you're going to form. And then we form the polymer gel network. And then after a chemical processing step that is used to overcome the binding forces in the tissues, we add water. So the polymer expands and the biomolecules expands and separates out and expands in space from each other. And so, as you can see, since the resolution is directly related to the expansion factor, if we increase the expansion factor, basically the resolution improves. So that means that if we do this process iteratively, that is after first round of expansion, if we can form another hydrogel and expand again, the brain tissue will expand more and more, and that means the resolution will improve. And in the first version of this technology, which we call the Iterative Expansion Microscopy and Iesm, what it did was that we used DNA conjugated antibodies to anchor the biomolecule of interest to the hydrogel network. And after the first expansion, the biomolecules were actually discarded and the DNAs were used as a proxy to hold the location information of the biomolecules. Now, this technology had some limitations. First of all, we could not retain the biomolecules themselves, and we were using DNA as a proxy. And moreover, we had to create these DNA conjugated antibodies for those. We cannot use the conventional off-the-shelf antibodies that are available easily. And another issue, and this is not only for IEXM, but for any super resolution technologies, is that when we use antibodies to identify a biomolecule of interest, the antibody, the primary and the secondary antibody itself has about a length of 20 to 30 nanometer. So that involves an inherent error just due to the presence of these antibodies. So we cannot go much, we cannot do much better than 30 nanometer. And another shortcoming was that IEXM, it was compatible with protein molecules, but RNA was destroyed in the chemical procedure that was used. So to overcome this process, we developed a technology which we call expansion revealing. We say it revealing, which I will discuss later on. It helps us to reveal some invisible biomolecular structures in the brain, which were previously not seen using other technologies. So we made some new discoveries, which I will go over briefly. So this expansion revealing technology, we were actually able to retain the biomolecules through all the steps of expansion. So it has the obvious advantages because we could do post-processing of the biomolecules. We do not need any DNA conjugated antibodies anymore, all off-the-shelf antibodies. And since now the biomolecules are retained through all the steps of expansion, we can bring in the antibodies after the expansion step. So the effective size of the antibodies will be reduced by the expansion factor. So if we achieve about 20-fold expansion and the size of the antibodies are 30 nanometer, their effective size becomes 30 divided by 20, around 1.5 nanometer, which is basically negligible. And another great advantage of doing the antibody staining after the expansion is this novel decrowding effect that we get. So suppose there is a biomolecule here, the red one, that we are trying to stain with antibodies. So before the expansion, the red biomolecule is basically in a crowded environment surrounded by many other biomolecules. So this antibody cannot reach and target this molecule. However, after we do the expansion, we have this decrowding effect and all the biomolecules get separated from each other. And now this antibody can go and target our biomolecule. So actually that helps us to visualize some unseen structures in the brain, which could not be visualized prior to expansion. And this process is not only compatible with proteins, but also with RNA and DNA. Basically anything that you can functionalize and attach to the polymer network can be seen with high resolution. And so we started by looking at some stereotypical structures in the brain, and one of them is microtubules. So microtubules are actually very interesting structures. You can think of them as pathways on which there are actually motor proteins that walk on top of it. So it was very interesting coming from an electronics background, coming to biology, when I came to know there are actually protein molecules which have like similar to our legs, they have a functionality, and within our cells, they are actually walking around on these pathways called microtubules. And these microtubules have around 20 nanometer of diameter, which you cannot resolve using a conventional microscope. But after expansion, you can see that we can resolve these sidewalls. And so we wanted to also employ this technology to, synaptic structures. Synapse is a region through which one neuron in the brain talks to the other neuron. And there are many biomolecules and synaptic proteins that occur in a crowded state there, and their organization and architecture actually defines the brain structure. So we wanted to apply our technology to synapse and understand more about their architecture and their functionality. And so this is an image of a single synapse that you see here. And you see two protein molecules here in red, PSG95, and boson in green. If you see that without the application of EXR, they look like an overlapping blob. But after you apply EXR, you can actually begin to see some structure in these two, and you can actually separate out these two proteins. And between these two proteins, what you see this gap is actually the synaptic cleft. So basically, these two proteins are transsynaptic proteins. That means this PSD95 is in one neuron here, and the boson is in the other neuron, and they're connected through this synapse. And there is this gap between these two, which is called the synaptic cleft. And that is exactly what you see here, which cannot be seen, say, without the resolution. So now that we know that we can actually resolve and get the architecture of the synapse, we wanted to see what kind of biological questions we can answer with this technology, and what we can learn about our the brain function. So I'm not a biologist myself, so we collaborated with Professor Thomas Van Platt at University of Maryland, and figured out it would be very interesting to see the configuration of a few different proteins in the synapses to start with. And so before I go into that, I will explain briefly about the synaptic function for our audience who might not be working related to this field. Suppose there are two neurons here, one shown in blue and another in green. And in here, the blue neuron wants to talk to the green neuron. So when this blue neuron wants to talk and it becomes active, what happens is that the potential inside this blue neuron will increase. And there are voltage gated calcium channels which will open up as the potential here increases. And then calcium ions flow in. Now, as the calcium ions flow in, they will actually lead to the fusion of these vesicles, which contains these neurotransmitters. And that's shown in magenta here. Now, as this fusion happens, these neurotransmitters are released, and then they travel through the synaptic cleft, and then they go and get attached to the receptor molecules, which are in this second neuron, which is this green neuron. And then as these receptors, they get attached to these neurotransmitters, as a response to this attachment, these receptor molecules will open up. And then based on the type of the receptors, they will let in either positive or negative ions flow in. So that can either increase or decrease the potential of the second neuron. So that is how, if one neuron wants to talk to the other neuron, it can actually affect the second neuron through this whole procedure of calcium ion flowing in, fusion of this vesicle, release of the neurotransmitters, and then them going and attaching to the other receptor molecules. So we thought that to start with, we would look at these three molecules. One is RIM, another is PSG95, and another is the calcium channel. So RIM is actually a protein which dictates where this neurotransmitter is going to be released. And PSG95 are protein molecules that act as a scaffold for these receptor molecules. So we wanted to see whether are these proteins just randomly oriented, or there is some kind of architecture that helps in its functionality. So we applied our technology to mouse brain tissue, and what you see here in left is the image of a single brain synapse, and the one shown in red is this protein rim, and in blue is the PSD95. And we also characterize basically the distance between the rim and the PSD95 molecules, which comes out to be around 60 nanometer, which seems to be in the right direction, because that's what people have found through, say, electron microscopy and all. So we try to analyze our the synapses in further details, and without going into the details of the analysis, so the coming to the results, what you found is something very interesting. We found that these biomolecules in the synapses, they are not just uniformly and randomly distributed. They actually form nanoclusters, and these clusters are around 60 to 80 nanometer in diameter. What is even more interesting is that nanoclusters of one protein, they sit directly above the nanoclusters of other protein. For example, nanoclusters of rim, the center of these nanoclusters align with the centers of the other protein PSD95, and that is with nanoscale precision. We're not even talking of micron scale precision, but the centers align with nanoscale precision. So this is something very interesting. If you remember, rim and PSD95 are in two different neurons. One is the one that is trying to talk to the other, and PSD95 is present in the neuron which is getting influenced. So these are in two separate neurons, but they have nanoscale precision in them. So why does that nanoscale precision happen? And what does that tell us about the brain functionality? Because we were very intrigued by these findings. So what happens is that, as I mentioned, that rim is a protein which dictates where the neurotransmitters are going to be released, and PSD95 acts as a scaffold for the receptors. The fact that the neurotransmitter are released directly above the point where the receptors are clustered, that reduces the distance that these neurotransmitters have to travel, because if these receptors are just randomly oriented, they had to travel longer distances, and then their probability of getting lost would increase. So by clustering receptor molecules directly above where the neurotransmitters are going to be released, Mother Nature has actually optimized the pathway of these neurotransmitters and made this process very efficient. Now, if we think of the tunnel field effect transistors we were designing, we were trying to reduce the barrier width to atomically thin dimensions so that we can increase the tunneling efficiency. And in our brain, Mother Nature has actually designed the architecture of biomolecules such that it can increase the tunneling efficiency of the neurotransmitters, and that directly increases the efficacy of synaptic transmission and helps to make our brain computation more efficient as well. So as nanoelectronics researcher, we always worry about, like nanofabrication, nanopatterning, how we can align with the high precision. But however, when we come to know that in our brain, Mother Nature has designed this kind of architecture so that it can improve and increase the efficacy of brain transmission, synaptic transmission and brain function, that really blows our mind. And again, as I said, this calcium ions flow through, and then that actually helps in the fusion of this synaptic vesicle. So we found that this calcium channel are actually also clustered near this fusion site. That also increases the efficacy of calcium going and help in this fusion of the synaptic vesicle. So we are actually currently using this technology to look into not only healthy brains, but different kinds of neurological diseases like Alzheimer's, Parkinson's, and autism. And to just have a brief overview of what we are doing, showing an example from an Alzheimer's brain. So this is Alzheimer's model for mouse. And there actually what we find is that so there are these misfolded proteins that are formed. They are extremely toxic in nature. These are called amyloid beta that are formed in the Alzheimer's brain. And we often think that any kind of disease is related to disorder. We call it neurological disorders. But what we found something interesting is that in Alzheimer's brain, these amyloid beta proteins actually form nanoscale periodic patterns in a very ordered structure. As you can see here, this amyloid beta nanostructures, and you do some analysis, it seems they have periodic patterns that they present. And what is interesting is that even if we look at the same level of resolution, but without doing expansion revealing technology, these nanopatterns are actually not visualized. So that is why this technology, as I mentioned, we call it expansion revealing, because it helps us to reveal nanoscale architecture, which cannot be seen without this expansion procedure. And so how much can we expand? We can actually expand 100-fold. So you can, so what you see here is a 50-fold expanded cell body, and this is a 65 expanded dendrite, and then another 100-fold expanded cell body. So we can actually go to higher levels of expansion to increase the resolution even further. So to quickly summarize, I actually showed you two completely different technology. One is a transistor, which helps us to overcome this fundamental limitation in power and dimensional scalability. And another technology which helps us to decipher the biomolecular building blocks and tells us about how brain makes our synaptic transmission and neuronal information processing more efficient. Though completely different technologies, I also talked about the synergies in terms of nanoscale precision and tunneling that happens between these two technologies. And in our lab, actually, we are trying to fuse these two fields together. So we call our lab nano cybernetic biotrek. So nano because we are dealing with nanoscale materials like atomically thin materials and nanoscale devices. Cybernetic because cybernetic in a broad term means the use of technology to control systems. In our case, the use of technology to either build new kinds of electronic systems or technology for modulating and understanding biological structures and biological systems. So biotrek, bio is for biology, and trek because research for us is an adventurous scientific voyage. And in this voyage, you are fusing engineering, applied physics, and biology together. And our group's two major research directions are inventing disruptive technology for nanoelectronic computation, specifically low power energy efficient devices, and then fusing these new technologies with biology to create new paradigms for life machine symbiosis. And a few projects that we are doing in our lab right now. So these are all experiments, ongoing experiments. I hope in a year or two I will be able to tell you in more details about this. One is life machine symbiosis. You are basically fusing nanoelectronics with biology to create new biohybrids. And biohybrids of fixing, basically you can think of cyber tissue, mixing nanoelectronic devices with biological tissues, both for recording as well as for therapeutic modulation. And also we're developing new kinds of nano implants for energy harvesting and wireless sensing, specifically from the brain. And we also continue to push forward the boundaries of low energy, low power nanoelectronic interfaces, including using quantum mechanical devices and nanomagnetics. So we have actually a lot of openings in our group, and for both students as well as postdocs. We are actually open to enthusiastic students irrespective of the disciplines. Some of the specific areas would be for students who are interested in, say, electromagnetism and in RFID design for energy harvesting and applications in brain. Students interested in nanomagnetism, magnetoelectrics, nanoelectronics, applied physics are also very welcome. We have actually a postdoc opening in our group, which is like currently open, and we have put up this position, this post in several avenues, so that this is specifically for brain stimulation and recording. Basically, we are developing these devices that we nanoelectronic devices that you want to implant, and we would like to have someone, a postdoc, might be interested in configuring these devices and have experience in biological experiments, specifically with mice, because that's the animal model we are using. So again, while these are the specific areas, you are actually open to enthusiastic and passionate students across different other disciplines. And I believe someone who wants to learn and very enthusiastic and passionate, they can actually excel in any field, irrespective of their specific background. So yeah, if you're interested, shoot me an email. And the email address is given here, and you can also find more details about the openings and in this website that is given here. So finally, to acknowledge all the wonderful people, my students, collaborators, and colleagues that I have worked with, without whom any of the work would not have been possible. And also a huge shout out to my family members who have always been very encouraging and inspiring. And actually, some of them, I believe, are listening to this talk today. And finally, thanks to you for your patience and being here with me today. Thank you very much. So I guess I will take questions.
Yeah, so it looks like we already had one question in the Q&A from Saraj Kant Singh. I can promote them to ask.
I think the question is, has a quantum amplifier solution been researched rather than thinning the barrier? I'm interested if quantum acoustics can be used to couple with the thermal oscillations at quantum level. Oh, yeah, that is a very interesting question. So for the transistor design that I was exploring, I didn't really explore any amplification of this, quantum tunneling effect to other acoustic waves or anything. Partly because in electronic devices, we wanted to modulate basically the barrier height and width, applying voltage. So thinning down the barrier height and lowering the barrier was obvious choice with voltage, but it's very interesting idea, and it might be possible to come up with new ideas and new ways where you can couple the photoelectric device and use acoustic waves for tunneling. Yeah, I'm not sure, but I'm sure that I'm not sure of the exact technology how you will do it, but definitely there could be some interesting things to think about in this direction.
I have a general question. Great talk, by the way. It was really interesting.
Thank you.
So I guess, thinking about your PhD work versus your postdoc work, in electronics, electrons are what's communicating information across these transistors, whereas in the brain, it's these ionic forms of communication with like calcium ions, for instance. I wonder if you can just comment on the differences between the two, and does that give some evidence to potentially why the brain is such an efficient or low power computer, so to speak?
Oh yeah, actually they are, we know very little about the neuronal information process in the brain, but we have some clues as to why the brain is more efficient than the electrons. So if we think about it, the speed of electrons is so much higher than the brain. Oral electronics can easily work in gigahertz. However, our brain itself is actually slow. Its computation speed, like maybe tens of hertz, kilohertz maximum. So speed-wise, brain is slower, but still it's just so much energy efficient. It can do very complex computation. So some of the reasons, one reason is that if you think of an electronic circuit, the number of connections are minimal. However, in the brain, one neuron basically can connect to 10,000 other neurons, and so this connectivity is high. So that increases the functionality and computation efficiency a lot. Second thing is that in the electronic circuits, how we have designed, we have this problem that the memory and the computation are separated from each other. So it takes a lot of time to basically move back and forth between the computational elements and the memory. In the brain, however, memory and computation are kind of linked to each other through the synapses itself. So that makes it very efficient. And few other things also are that in the brain we have basically we have there is error, but because we have different pathways, there is a lot of redundancy. However, in our electronic devices, basically we make that we have designed it such that it is relatively error-free, but that also leads to a lot of the power that we consume. So if we can increase the redundancy, but make each of the individual computations maybe induce some error in that, maybe we can reduce the computation as well. Moreover, our electronic circuit is synchronous. There is a clock, and whenever we apply the clock, only then whole computation everywhere happens. I mean, in brain is basically in each, basically there is no like central clock that is present, and whenever the signal comes in, they can be transmitted. So in one way it is asynchronous if we think about it. So yeah, there are some very fundamental differences between brain and electronic computation, and actually we are learning from brain and like kind of implementing some of these in our electronic devices so that we can improve on our computational side as well.
Great. Any other questions from the audience? Please don't hesitate to type it into the chat or the Q&A box. Oh.
I see a hand.
Yeah, Deblina, it's quite late in India, but your topic was so interesting. I said, let's wake up in the early VRs, okay? So, I was really interested in the work you're doing. And however, when we take a reductionist view of, simplifying or, modifying the brain, and as you said, we expanded it and saw what kind of, patterns are there, we actually lose some of the synergetic things that come from complexity. Suppose, for example, you take a Kolkata minibus, you place very arranged people and people sit in the each of the chairs, then it's still there's capacity. And so because people are in a rush, they all fit into that same bus, and maybe 20 people go instead of just 5 inside the bus. So maybe nature has its own way of making it more efficient. And the moment we design things like transistors or cities, where we plan it and you put it, people place things in the layout, we bring rigidity. So don't you think we should have a systems approach rather than a reductionist view? That is a thought I had, so that maybe when we simplify things, we lose a lot of its synergies, and maybe in that process you come up with a thing which is more rigid and less efficient.
Yeah, I think that's a very interesting question. So I believe that, I think both these pathways are necessary and depending on the problems in hand. So in certain cases, so of course, in our, in computations and electronic devices and whatever research different groups are doing, I think in our research as well, it is possible to introduce this, random variables and, see that what the randomness brings into and what they have to offer. And then in certain cases, we have, when you want to understand the physics and design things well, we'll have to go through this, well-designed process. So nature had, billions of years to design things through, trials and errors. I believe that, through understanding and through, design of things well, we can do better than nature in less than in terms of time efficiency. But as far as complexity is concerned, we have and I like the idea of this systems approach, but I think there has to be both these pathways going together. Top down approach, where we have start from a system and try to simplify things to understand, and then also a bottoms up approach. I like it very much because when you simplify things and try to build things on top of it and then go to a more complex system, that basically enhances our understanding so that we can build and apply those knowledge to other things. So I kind of think both of them has to go hand in hand.
Thank you.
We have another question from in the chat. I can read it. It's, are you mostly concentrating on detecting neuronal diseases or also diagnostics? And how do you make sure when placing a nanochip in tissues, the insulation is correct in terms of fabrication? And are your devices toxicity free?
Yeah, so basically, the research you're doing can be divided into two main categories. One is reading out information from the brain, and another is writing information into the brain. So reading information is basically recording brain activity, and that is for understanding fundamental computation in the brain, but also for neurological diseases. For example, in different kind of neurological disorders, there's brain functioning and those things, you see some activity which is different from in healthy brains. And also when you want to correct those systems, we want to basically apply and write in information, modulate those brain functionality, electrical functionality through electrical stimulation is what we are focusing right now. So yeah, to answer your question, yes, both detecting signals, but also modulating them for therapeutics. And I think the second one is how do you make, the nanochip, the installation is correct in terms of fabrication. Yeah, I think those are something, there are different kinds of insulation that, that are there, that is possible. So some of them, so first of all, we basically do our experiments in vitro in air, and then we do it in say in solution in PBS. And there are also ways we can put it inside the brain and retrieve the devices after some time. So these are testing procedures, and we basically insulate our devices with different kinds of materials, like I think silicon carbide or silicon dioxide, and even polymer layers we have used. So based on the functionality that we want in our devices. So some devices, we want them to be transient, that it works for some time, and then, once that device is not required, they can dissolve in, but others we need for more chronic applications. So based on them, we coat our devices with different biomaterials. And as far as toxicity free, so nothing is completely free of toxicity, but what has been shown is that if you reduce the device size, because that makes it less and less invasive in nature. Our devices are not completely non-invasive, but we call it minimally invasive, because we are developing devices which are the sizes of few microns. The tissue displacement is very low. Definitely, much less invasive than electrodes that we put inside the skull. And we look at, glial response and inflammatory markers and see that those effects are also minimal. So these devices you're making are actually minimally invasive in nature.
I guess I have another question, or I guess more of a thought, kind of related to the discussion around controlling things precisely from the bottom up. I thought it was really interesting how you shared that there was that nanoscale precision in alignment at these synaptic junctions between these different proteins. And brains are plastic, and new synaptic connections are able to form and reorganize. So to me, that would imply that there's some sort of self-assembly mechanism where these really ordered, precise structures can form. And I wonder if there's any way to maybe design electronics that also are capable of like self-assembled optimization in the same kind of way. Is that something that you've thought about before or have any ideas about?
Yeah. So that's very interesting. So I think I would like to comment on one thing that you said about the plasticity. Yeah, so that's another aspect which our electronics currently misses. We basically have rigid structures all connected to each other, and they just work the same way. So there has been a lot of research that's going on basically to bring in this plasticity effect in electronic devices. Basically call it neuromorphic computing. And the way actually there are many different ways people have approached this problem, and specifically in ways where to emulate this short-term plasticity or long-term plasticity, or the synaptic weights that changes with there's neurons that fire together, wire together, those kind of concepts. And some of the ways people have done it is, suppose you have say 2 electrodes and separated by a dielectric, and in this dielectric you have some metallic nanoparticles. And if you continue to apply pulses between these 2 electrodes, initially you will have a high resistance. And however, if the number of spikes increase, what happens is, due to the heating effect, those nanoparticles melt and they form a bridge between the 2 electrodes. And then the connection and the resistivity between those 2 electrodes decreases. So that means you can think of, and the neurons that fired together for a while, they are kind of now wired together. So basically, now their resistivity has decreased. So there are a lot of novel materials like phase change materials, membristors, magnetic devices, with which people have actually implemented those kind of neuromorphic designs, and have brought in the plasticity, not through like physical change of locations, but through novel electronic ideas. And another thing I would want to say about in the brain itself, as you said, like this biomolecular organization changes with plasticity. And that is something also is very interesting. And what we found is that when we stimulate the brain, and in terms of long-term plasticity, when the connections between these neurons are supposed to be enhanced, this nanocluster, their size actually increased, and there is more basically connection between these 2 neurons when the connection has to be improved. And is the opposite when you have the connection is broken, or the plasticity, the reverse of that happens. What happens is that then the molecules becomes more diffuse, and this nanoscale alignment at that point is actually broken. So that's another way of basically inducing plasticity.
Very interesting. I saw briefly a hand was raised in the attendee list. Is there someone else who wanted to ask a question? Don't be shy. I guess I have one more question then. Not really a technical question, but the work that you've done is so different from each other. I mean, what was the learning curve like going into this neuroscience space? And was there things that you could draw from your PhD experiences that you think maybe could actually help you or gave you some unique perspective? Just interested to see how you tackled that challenge.
Well, yeah, it was a steep learning curve. Yeah, when I joined, when I moved to this new field, it was like I was completely unaware of the jargons. And like I have never held a mouse before in my hand. And all my work before was with, like crystalline hard devices. And then you're moving to soft tissue and gels. And I know initially in the lab when I started, I was like vortexing antibodies, and my colleagues were saying, no, they are really sensitive. You should be very careful because you should not be doing those kind of things. So the learning curve was very steep. I think I enjoyed a lot, and that was by design. I had thought about whether I really want to go to a new field, and because that field itself it's like learning from the very scratch at that point. So yeah, it was challenging, but it was also exciting. And the reason I went that route, because I felt that once I have this experience in my own group, I can actually fuse these two together and build something unique. So that was a motivation. So I think that was exciting overall, I would say.
Absolutely. I think that's how really innovative stuff gets pushed forward, is by bridging these kind of disparate fields and expertise. All right. Any other questions? Last call. Okay, well, in that case, let's once again thank our speaker for the amazing talk. And yeah, another round of applause.
Yeah,
thank
you
so much
for organizing this amazing,
Thank you, Prof.
Yeah, thank you.
Thank you.
Okay, thank you, everyone. So our video will be uploaded to the website soon. Keep tuned. Okay. So thank you, Vivian.
Yeah, thank you, Vivian.
Thank
No problem. See you guys.
See you.